Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. In this lecture, I will be explaining you nucleic acid structure and organization. Let's get into the details. The central dogma of molecular biology is storing the genetic information and transforming that genetic information into RNA and then into protein to change the physiological function of a cell. Now, an organism must be able to store and preserve its genetic information and pass that information along to the future generation and express that information as it carries out all the process of life is the central importance here. So, the gene expression encodes the information into an mRNA, and information in the mRNA is decoded into a protein molecule, and then a protein molecule changes the physiology of the cell, and that is what is the central dogma here for a molecular biology. Alright, so classically a gene is a unit of DNA that encodes a particular protein or an RNA molecule. Now let me show you the comparison between a gene expression and a DNA replication. So gene expression, it produces all proteins an organism requires, whereas the DNA replication, it duplicates the chromosomes before cell division. Gene expression, it causes transcription of DNA, where RNA copy of a small section of chromosome, like average size of human genome, which is 10 to the power of 4 to 10 to the power of 5 nucleotide pairs. So, whereas the DNA replication, it copies the entire chromosome, that is the average size of human chromosome, which is 10 to the power of 8 nucleotide pairs. So, it is copying everything there in a DNA replication. And in gene expression, transcription occurs in the nucleus throughout the interphase, whereas in DNA replication, it occurs only during the S phase. So, that means DNA replication happens during the synthesis phase or the S phase of cell cycle. Now, the gene expression, it causes translation of RNA, that is the protein synthesis, which occurs in the cytoplasm throughout the cell cycle, whereas the DNA replication, it happens in the nucleus. Now, let's look at eukaryotic cell cycle. The concept of cell cycle can be used to describe the timing of some of these events in a eukaryotic cell. The M phase is the mitotic phase, where the cell divides into two. Whereas the interphase, it is the time between two cell divisions or mitosis. Furthermore, interphase is divided into G1 phase, which is also called as gap 1 phase, and that is the preparatory phase for DNA synthesis. And then we have S phase or the DNA synthesis phase, where the replication of DNA occurs. Whereas G2 phase or the gap 2 phase, it is the preparatory phase for the cell division. And also there is a G0 phase, as it is shown here. G0 phase is seen in cells which have stopped dividing and are in this phase. That is, example, we have muscles and neurons which are in G0 phase. Some of the drugs will target cell cycle. In that, say, S phase targeting drugs are methotrexate, we have 5-fluorouracil, hydroxyurea. G2 phase targeting drug, we have bleomycin. M phase targeting drugs, we have paclitaxel, vincristine, vinblastine. And non-cell cycle specific drugs, we have cyclophosphamide and also cisplatin. So, these are some of the drugs that will target different phases in the cell cycle. Alright, now let me explain you nucleotide structure and nomenclature. So, 5 carbon sugars that are present in the nucleic acids are ribose in the ribonucleic acid and deoxyribose in deoxyribonucleic acid. Bases are nitrogen containing molecules. We have 2 types of bases. One is the purine base and other is the pyrimidine base. And then DNA has adenine, guanine, cytosine and thymine. These are the bases present in DNA. Whereas RNA has adenine, guanine, cytosine and uracil. So, the difference here is in terms of bases. See, the adenine and guanine, these two are the purine base, as it is shown. The structure of adenine and guanine is shown here. So, Purine base, it contains 9 atoms, and the numbering in the purine will go in anti-clockwise manner. So I'll number that as 1, 2, 3, 4, 5, and 6, 7, 8, and 9. So these are the 9 atoms in a purine ring, and there is an adenine. In adenine, there is an amino group attached to the 6th carbon. So that's an adenine. Similarly, we have a guanine. So adenine and guanine, these are purine bases. While pyrimidine bases, we have 3 of them. That is cytosine, uracil, and thiamine. So there are 6 atoms in a pyrimidine ring, which goes in a numbering here. It goes in a clockwise manner. So in DNA, we have 2 purines and 2 pyrimidines. So purines are adenine and guanine, and pyrimidines are cytosine and thiamine. Similarly, in RNA, purines are adenine and guanine, just like DNA. Whereas the pyrimidines, cytosine is same, but the uracil, in place of thiamine in DNA, there is a uracil here. So now the sugar in DNA is a deoxyribose, whereas in the RNA it is ribose. The difference here is the second carbon of ribose is hydroxylated in RNA, whereas it is hydroxyl group is removed in DNA. That's what makes it as deoxyribose there. Now, the nucleosides, coming to nucleosides. So we have seen the bases. Bases are adenine, guanine, thymine, cytosine, uracil. Those are the bases. Now, what are nucleosides? Nucleosides are base plus ribose 5-phosphate or deoxyribose 5-phosphate. These are the bases. So, base plus sugar, and simply write it as base plus sugar is the nucleoside. Okay, so pentose sugar and the base. Whereas nucleotide is base plus sugar, additionally there is a phosphate. So that's the difference between a base, nucleoside and nucleotide. So examples for nucleotides given here: uridine monophosphate, it's a nucleotide, and then deoxyuridine monophosphate, and then adenosine. Adenosine is a nucleoside. Adenosine is a nucleoside. It's not phosphorylated. Similarly, we have deoxythymidine, which is a nucleoside with the sugar which is in deoxy form. So that's how the nucleosides and nucleotides are differentiated. You'll have to look for the additional phosphate in nucleotide compared to nucleoside. Now, here is the table that will give you all the base, nucleosides and nucleotides for both DNA and RNA here. So, adenine, adenosine, adenosine monophosphate, diphosphate, triphosphate, like that for all other purine bases will go on. Guanine, cytosine, uracil, thymine. So, it just goes on like that. Only thing is, you don't need a deoxy form of uridine triphosphate for DNA because it is in the form of deoxythymidine triphosphate there. Coming to nucleic acids. Nucleic acids are polymers of nucleotides joined by 3' 5' phosphodiester bond. Remember that. So the 3' 5' phosphodiester bonds are the ones which are joining two nucleotides together. So that is the phosphate group which links 3' carbon of a sugar to the 5' carbon of the sugar that is coming in. That's the next sugar which is there in the chain. That's what is the 3' 5' phosphodiester bond. So each strand has got a distinct 5' end and 3' end, and thus it has got a polarity. So a phosphate group is often found at the 5' end and a hydroxyl group is often found at the 3' end because that's how they are attached. Sugar, a ribose 5-phosphate, as let me show you that here. So look at the 3' 5' phosphodiester bond here. Here is the 5' phosphate, the 5th carbon attached with the phosphate, and that one, it is now the 3' end is here, so which is attached with another phosphate, that is the 5'. So basically we have 5' end here, and we have the hydroxyl group which is free there, that is a 3' end. So this strand, it runs in 5' to 3' direction. In the same way, so look at this 5' end this side and the 3' end is this side. So it goes from 5' to 3'. So this type of directionality is there in whenever nucleic acids are made with the 3', 5' phosphodiester bond, and thereby there is a polynucleotide chain is made, and that's how the nucleic acids are synthesized, and they can base pair with each other depending on, so whether it is a double-stranded DNA is made or double-stranded RNA is made and all that. So the base sequence of nucleic acid strand is written by convention in 5' to 3' direction. Remember that. And that is going from left to right. That's what is the 5' to 3' end. So according to this conversion, the sequence of the strand on left must be written as like 5', and then if the sequence is like 𝗠𝐶𝐴G, and then you write it as 3'. If you are going from 5' to 3' end, so it will be thymine, cytosine, adenine, adenosine and guanosine. So that's how you go there. So TCAG. So if you are writing in a reverse order, so that end must be, you have to start from 3' GACT 5'. You have to go in that way, so that it exactly indicates how the sequence is running. So in eukaryotes, DNA is generally double-stranded DNA, which you write it as dsDNA, that indicates double-stranded DNA. And RNA is usually a single-stranded RNA. ssRNA means single-stranded RNA. This is what happens. So in eukaryotes, the DNA is usually having double strand, and RNA is usually having a single strand. So certain exceptions can occur in viruses, because some of the viruses will have a single-stranded DNA genome, and also some of them will have double-stranded RNA genomes also. You just need to have an idea on that. That is in the viruses. Now let's look into DNA structure. So some of the features of double-stranded DNA includes: the two strands are anti-parallel, that means they run in opposite direction in a DNA. That means one strand is running in 5' to 3' direction, other strand is running from 3' to 5' direction. That's first point there. Second thing is, these two strands are complementary. That means always adenine will pair with thymine, and usually that is done with 2 hydrogen bonds, whereas guanine pairs with cytosine, that usually happens with the 3 hydrogen bond. Thus, the base sequence on one strand defines the base sequence on the other strand. It will be like this. In general, if I write, say, adenine, cytosine, guanine, thymine, something like that, then I know what is the base sequence in the other strand. So if I am running this particular strand as 5' to 3' direction, so other strand which is complementary to this strand, so it will be double bond thymine 3' that side, then triple bond cytosine with the guanine there on the other side, triple bond then cytosine on the other side, double bond adenine on the other side, and then this will be 5'. So like this, once you know a base sequence in one strand, you exactly know the base sequence in the opposite strand. Only thing is, it is running in the opposite direction. And also, for this particular base sequence formation or the base pairing, it will follow something called as Chargaff's rule. Chargaff's rule says that the amount of adenine equals to the amount of thymine, and also the amount of guanine equals to the amount of cytosine. Thus, the total purines will be equal to total pyrimidines. That's what is the Chargaff rule. Because if you count here, for every guanine, there is a cytosine. So, on either side, this is how the percentage of the percent purines equals to percent pyrimidines. That is what a Chargaff rule says. And most DNA occurs in nature as a right-handed double helical molecule, which we call it as a Watson-Crick DNA or a bDNA. Now, the hydrophilic sugar phosphate backbone of each strand is on the outside of the double helix, and the hydrogen bonded base pairs are stacked in the center of the molecule where they make a hydrogen bond like adenine, thymine, guanine, cytosine. So, there are about 10 base pairs per complete turn of one helix. A rare left-handed double helical form of DNA that occurs with the guanine cytosine rich sequence is known as a zDNA. The biological function of zDNA is unknown but may be related to gene regulation. Now let's look at denaturation and renaturation of DNA. A double helical DNA can be denatured by conditions that will disrupt the hydrogen bonding and base stacking, and this particular process we call it as melting of double helix DNA into two single strands that separate from each other. No covalent bonds are broken in this process. Like heat, alkaline pH and chemicals such as formamide and urea are commonly used to denature a DNA molecule. Denatured single-stranded DNA can be renatured or annealed if the denaturing conditions are slowly removed. For example, if a solution containing a heat denatured DNA is slowly cooled down, the two complementary strands can become base paired again. And such renaturation or annealing of a complementary DNA strands is an important step in a process. And such renaturation or annealing of complementary DNA strands is an important step in probing a southern blot and also in performance of polymerase chain reaction, where hybridization is necessary. So this is what is something about renaturation and denaturation process, where a double-stranded DNA is denatured like use of heat, which will separate hydrogen bonding process, and a single-stranded DNAs are made. So when the cooling is done, there will be re-annealing process or renaturation of two single-stranded DNA to become a single double-stranded DNA. So now let's get into organization of DNA. Large DNA molecules must be packaged in such a way that they can fit inside the cell and still be functional. So there is a concept called as supercoiling. So mitochondrial DNA and DNA of the most prokaryotes are closed circular structures. These molecules may exist as relaxed circles or supercoiled structures in which the helix is twisted around itself in a three-dimensional space. Now, the supercoiling results from strain on the molecules caused by under or overwinding of the double helix. So, there is a negatively supercoiled DNA which is formed if the DNA is wound more loosely than in Watson-Crick DNA. This form is required for most biological reactions. So, negatively supercoiled DNA is a more loose DNA than the Watson-Crick DNA, and it helps in the expression of genes, thereby transcription is going on and mRNA is made. So, whenever there is a transcription needed, whenever a gene has to be expressed, at that time there will be negative supercoiling will go on where the DNA is loosened, and the part of that gene wherever it is loosened, it will be expressed to undergo transcription process to give mRNA and mRNA into proteins. Now, the other process is positively supercoiled DNA. So, this is formed if the DNA wound more tightly than Watson-Crick DNA. Now, there are enzymes like topoisomerases. These are the enzymes that can change the amount of supercoiling in a DNA molecule. So, they can transiently break a DNA strand by alternatively by breaking and resealing the sugar phosphate backbone. So that's what is the function of topoisomerases. So whenever the supercoiling need to be changed, so topoisomerase enzymes will function. Now coming to nucleosome and chromatin structure. Nuclear DNA in eukaryotes is found in a chromatin associated with histones and non-histone proteins. The basic packaging unit of chromatin is referred as a nucleosome. Now, the histone molecules which are part of the nucleosome, these are rich in lysine and arginine, and because of the positive charges that are present in lysine and arginine, they will confer a positive charge on the proteins. Now, 2 copies of each of histones, that is H2A, H2B, H3, H4, will aggregate to form what is called as histone octamer. Now, DNA is wound around the outside of this histone octamer as it is shown here in the figure. So, the DNA, it is wounded around the histone octamer. It's like a ball and you are tying a thread around it, and imagine that the thread is the DNA and the ball is the histone which has got 8 the pairs of H2A, H2B, H3 and H4. And so that's what it is explained here. Now, the DNA wound around outside of this octamer to form a nucleosome. So, nucleosome, these are the series of nucleosomes, sometimes referred as beads on string, but it is more properly referred as a 10 nanometer chromatin fiber. So, the histone octamer around which when the DNA is wounding around that particular structure, which is a beads on string appearance, if you collectively look at it, so that thing is referred as a 10 nanometer chromatin fiber. Now, histone H1 is associated with the linker DNA found between the nucleosomes to help package them into a solenoid-like structure, which is known as a 30 nanometer chromatin fiber. Furthermore, condensation occurs to eventually form the chromosome. So, like this, a 10 nanometer fiber with the H1 histone and connecting with another 10 nanometer fiber. So, overall making a 30 nanometer fiber, and further condensation will go on. Eventually, it will make a chromosome structure. And each eukaryotic chromosome in Z1 or Z0, it contains one linear molecule of double-stranded DNA. Now, the cells in interphase contain 2 types of chromatin. One is a euchromatin, which is more opened and available for gene expression, and the other is heterochromatin, and heterochromatin is much more highly condensed and it is associated with areas of chromosomes that are not expressed. So, that's the difference between euchromatin and heterochromatin. So, the figure here where the double-stranded DNA initially making a 10 nanometer chromatin and the beads on string appearance, all these things are coming together and making a nucleofilament. So, the nucleosome, which is 10 nanometer chromatin, coming together, all of them making a 30 nanometer chromatin, which is called as nucleofilament, and then 30 nanometer fiber forms the loops which are attached to the scaffolding proteins, finally making a higher order packaging, overall making a heterochromatin. So, the euchromatin is more loosely formed chromatin where the DNA expression, gene expression can go on, whereas the heterochromatin is highly condensed chromatin. Usually, genes here are not expressed. Now, the nucleolus is a nuclear region which is specialized for ribosome assembly. And the chromosome structure is visible during mitosis, because during mitosis here it is a highly condensed heterochromatin DNA is present, and chromosome analysis is done on mitotic chromosome because it is highly condensed there. A karyotype analysis is usually done on a metaphase chromosome, and the banding techniques are used over a prophase or a metaphase chromosome. So, remember, these are the stages of chromosomes where different techniques can be used. So, this is all about nucleic acid structure and organization. Thank you.